A very good day to Dr. Rosmawati. Today, my group, Group 1, Subgroup 6, will do a special presentation on the usage of LYSOZYME in the pharmaceutical industry for the assignment 1. My group consists of 5 members. Me, myself, Petrus Luna, Mohammad Syahmi Danial, Mohammad Hamirul, Nurul, Aleeya, Mysara, and Rebecca Sherene. LYSOZYME is an enzyme that catalyzes the hydrolysis of beta-1,4-glycosidic bonds in the peptidoglycan layer of bacterial cell walls, leading to cell lysis. It is naturally found in various biological fluids such as tears, saliva, and egg whites, serving as part of the innate immune system. LYSOZYME was discovered by Alexander Fleming, the same scientist who later discovered penicillin. Fleming observed its antimicrobial activity when he accidentally contaminated the bacterial culture with nasal mucus, which led to the bacteria destruction. He identified the substance responsible as lysozyme, marking the beginning of enzyme-based antimicrobial research. In the decades following its discovery, lysozyme was extensively studied for its ability to break down bacterial cell walls. The enzyme was one of the first whose structure was determined by using X-ray crystallography in the 1960s by David Chilton Phillips and his team, providing critical insights into enzyme mechanism and structure-function relationship. In 2023, the global lysozyme market was valued at approximately 530 million USD and is projected to reach around 890 million USD by the year 2032, indicating a compound annual growth rate of 6.1% during the forecast period.
Lysozyme is sometimes called the natural antibiotic because it was one of the first enzymes discovered to have antimicrobial properties, breaking down the bacterial cell walls. It's so safe and effective that it's even used in baby formulas and eye drops to protect against infections. And did you know lysozyme was first discovered in tears by Alexander Fleming, the same scientist who discovered penicillin? And fun twist, lysozyme is used in cheese production as a natural preservative to prevent spoilage caused by harmful bacteria, demonstrating its versatility beyond pharmaceuticals.
In the pharmaceutical industry, lysozyme addresses several critical issues offering potential solution due to its antimicrobial properties, natural origin, and enzyme specificity. The first challenge is antimicrobial resistance, commonly known as AMR. The widespread misuse of antibiotics has led to the emergence of drug-resistant bacterial strains, posing a severe threat to public health. Lysozyme's natural ability to lyse bacterial cell walls provide an alternative or complementary strategy to conventional antibiotics, reducing reliance on antibiotics and mitigating resistant development. The second challenge is infection control in wound management. Bacterial infection in wounds at surgical sites can delay healing and lead to complication, especially with resistant strains. Lysozyme can be incorporated into wound dressing to prevent bacterial colonization, accelerate healing, and reduce infection rates. The third challenge is biocompatibility in drug delivery system. Traditional antimicrobial agents used in drug delivery systems may cause adverse reaction or lack specificity. Lysozyme is biocompatible and can be integrated into drug delivery systems, such as hydrogels or nanoparticles, to provide localized antibacterial activity with minimal side effects. Finally is the preservation and sterilization of pharmaceuticals. Contamination of pharmaceutical products such as eye drops, injectables, and vaccine can compromise safety and efficacy. Lysozyme can act as a preservative in formulation to inhibit microbial growth, ensuring the safety and longevity of this product.
Next is the application of enzyme in pharmaceutical industry. Firstly is antibacterial properties. Lysozyme is used in the formulation of adjuvant drugs that enhance the efficacy of antibiotics and analgesics. Also, it serves as a diagnostic tool for monitoring disease progression in humans and animals. Second, in drug delivery system, lysozyme acts as a stabilizing agent, preventing aggregation. This enhances the effectiveness and safety of this delivery system, while it also can enhance bioavailability by breaking down bacterial cell walls. Thirdly is cancer treatment. Lysozyme has demonstrated direct anti-cancer properties by inhibiting the growth of various cancer cells, including breast and melanoma. Next is hypertension management. Lysozyme can bind to angiotensin-converting enzyme ACE, which is crucial in regulating blood pressure. Lysozyme also influences immune cells, especially monocytes, which play a role in inflammation. Lastly, antiviral agents. Lysozyme can inhibit the replication of viral nucleic acid. It has been shown to enter cells and block the activity of polymerases, which are essential for viral replication. Lysozyme has shown effectiveness against various viruses, including herpes simplex virus and HIV. Next is specific regulation for lysozyme use in pharmaceutical within Malaysia. Firstly is drug registration. All pharmaceuticals products, including those containing lysozyme, must be registered with the National Pharmaceutical Regulatory Agency (NPRA) before they can be marketed. Second, Malaysian Medicine Act 1984, lysozyme products must comply with this act to ensure they meet safety and efficacy standards. Next is Good Manufacturing Practice (GMP). It ensures that products are consistently produced and controlled according to quality standards. For clinical trials, if it conducted to evaluate new formulation or application LYSOZYME, they must receive approval from the NPRA. Then, once LYSOZYME-based products are marketed, they are subject to post-market surveillance to monitor their safety and efficacy in real-world use. Lastly, international consideration. Malaysia is a member of the Asian Economic Community AEC, which promotes regulatory harmonization among member states.
LYSOZYME plays an important role in the pharmaceutical industry, particularly in antibacterial treatments and biotechnology applications. There are 6 steps in the lysozyme mechanism. For the first step, which is the substrate binding, in pharmaceutical, lysozyme is valued for its ability to bind to bacterial cell peptidoglycans, which are abundant in gram-positive bacteria. This binding initiates the process of breaking down these harmful bacteria, offering a natural defense against infections. The second step, the formation of the enzyme-substrate complex. Once bound, the enzyme positions the glycosidic bonds or the sisal bonds near its catalytic residues, which are GLUT35 and aspartate 52. This step highlights enzymes' efficiency as a biocatalyst, which is useful for creating antibacterial formulations or enhancing natural immune response in products like wound care, ointments, and nasal sprays. The third step, which is the cleavage of the glycosidic bonds. At this point, the enzyme breaks the glycosidic bond in the peptidoglycan structure. In the pharmaceutical context, this ability to disrupt bacterial cell walls makes lysozymes a potential adjunct in combination therapies to reduce bacterial resistance. The fourth step, which is the transition state stabilization, lysozyme ensure efficient catalysis by stabilizing the transition state. This property is key in pharmaceutical formulations where enzyme efficiency translates into effective bacterial elimination, especially in preservative systems or sterile environments. The fifth step, which is the hydrolysis of the intermediate, the enzyme facilitates the breakdown of its intermediate state, releasing sugar fragment. For the pharmaceutical industry, this highlights its role in safely and effectively degrading bacterial contaminants in various products, ensuring safety and compliance. The sixth step, the final step, which is the product release. Finally, lysozyme releases the cleaved bacterial components and regenerates itself. This self-renewing ability is particularly important in biopharmaceutical manufacturing, where enzymes like lysozymes are used to maintain the sterility of drug formations or as stabilizing agents in vaccines.
Lysozyme, often referred to as nature's antimicrobial enzyme, has gained significant attention for its diverse application in healthcare. Now, we'll explore the future opportunities and impact of lysozyme in the pharmaceutical industry. As antibiotic resistance becomes a global concern, lysozyme is emerging as a promising natural antimicrobial agent. Its ability to break down bacterial cell walls makes it an ideal alternative and complement to traditional antibiotics, offering a safer and more sustainable solution to fight against the infection. Researchers are increasingly exploring the lysozyme potential across various healthcare fields. Its applications include wound healing, immune system modulation, and innovative drug delivery system, highlighting its versatility as a therapeutic agent. And next, the biotechnology market is rapidly growing, with lysozyme playing a critical role. Its incorporation into pharmaceuticals, nutraceuticals, and biomanufacturing processes opens up a significant business opportunities as seen in the projected market growth rates. And lastly, ladies and gentlemen, another exciting prospect is lysozyme potential to reduce biomanufacturing costs. By improving the efficiency of production processes and serving as a sustainable alternative, lysozyme contributes to making biomedical innovation more accessible and cost-effective.
In conclusion, lysozyme is a crucial enzyme with significant applications in pharmaceutical industry. Its capability to break down bacterial cell walls make it valuable for various purposes, including antibacterial and antiviral therapies, drug delivery, and disease management. Additionally, lysozyme is used in food preservation and as a natural preservative due to its antimicrobial properties. With its increasing market potential, lysozyme continues to play an important role in advancing medical and pharmaceutical innovations. Researchers are exploring new formulations, and delivery methods, further expanding its applications. As the need for effective treatments grow, lysozyme's importance in modern healthcare will likely continue to increase.